Okay, welcome back. So what we're going to do today is a video lab. I know that's not the most ideal thing. You will still get to do part 2 of the lab, which is where you choose a substance and do a very similar process as this. So I'm going to do the lab for you today and then collect the data for you, and we'll see that tomorrow, and you guys can work on your pre-lab handout and do all that good stuff with the graphing, the tables and stuff there. Okay, so what are we going to do essentially? We do know that we're going to be using molasses and yeast. Yeast is a living organism, and in this pouch here, you can buy this at the local grocery store. It's called dry activated yeast. Dry activated yeast is not active until you put water or sugar with it, and then it starts to grow and divide and make more. So this is commonly used in bread and stuff like that for baking. Molasses is just a sweet syrupy mix, kind of very similar to like maple syrup and stuff, just a different product though, from a different plant. So ultimately what we're gonna do is figure out what concentration of molasses yeast grows best in. The way we're going to do this is with a serial dilution using these test tubes. So we're going to have test tube number 1, which is going to have the highest concentration of 100%, all the way down to the smallest concentration of 0.19%. So ideally, the yeast will grow in a certain concentration of molasses. The question is, which one? So that's going to be your hypothesis. List them in the order that you think is going to be the best, from 100% down to 0.19%. So the materials that we're going to be using: test tubes, molasses, yeast, as we talked about, a graduating cylinder to measure the molasses, little corks or stoppers to plug up the good old test tubes, and then we're also going to use DI water, which has no chemicals in it, no chlorine, fluorine, stuff like that, just pure H2O. We're going to use the veneer probeware right here and temperature probe to measure the temperature of some water I have over here heating up, because we've got to activate the yeast at the ideal temperature, and a little dropper to get the yeast out of there and put 3 drops into each test tube to get a good culture going. Okay, so those are the materials that we're going to be using for this particular lab. All right, so what I'm going to do first is going to take the molasses, and I'm going to pour 25 milliliters of it into our graduated cylinder. And I'm going to put it in test tube number 2. I'll explain why I'm avoiding test tube 1 later on. Okay, so we're going to start there, and it's going to be 50% of molasses and DI water. Okay, so I'm going to put 25 milliliters of molasses in here and 25 milliliters of DI water. I'm going to shake it up, mix it up thoroughly, and once I have a uniform solution, I'll pour it into test tube number 2. Then, with the remaining 25, I'm going to continue to add 25 DI water into it, shake it up, mix it, put it into test tube 2 or 3. And that will continue to get a dilution. So that'll be 50, 25, 12.5, and so forth down to 0.19%. Okay? The reason why I'm not doing test tube number 1 is because it's 100% molasses. And 100% molasses trying to pour out of a graduated cylinder is very difficult. So I'm going to do all of them, and if I do it correctly, I should have a pretty uniform line across the test tubes, so I can kind of accurately measure how much to pour directly in there. Spoiler alert here, the 100% usually does not produce any activity whatsoever from the yeast because it's just too sugary. So there's a little spoiler alert. You can add that to your hypothesis if you want. Okay, so I'm going to stop the video, and then I'm going to do a time-lapse video. All right. Okay, so now that I've got it all done, you can see the concentration from 100% down to 0.19%. Again, this is a serial dilution. As you can see, it gets the darkest to the lightest. So we have more water in this one, and we have more molasses down here. So the question is, which one of these is going to work best for the yeast to grow in? So that's what you're trying to make in your hypothesis. Now, the next thing we're going to do is add in 3 drops of the yeast from that little thing. I'm going to put 3 drops in there. And then I'm going to insert a tiny test tube, okay? So this little tiny test tube has to go inside this large one, upside down. And the reason for that is what we're gonna do is get the yeast to actually produce CO2. Why? Yeast, a living organism, does cellular respiration just like you and I do. As they break down sugars, they off-gas carbon dioxide. So we're gonna collect the carbon dioxide in this tube. Now, the tricky thing is you got to take this little tiny test tube and get it inside of this particular tube. I'm going to demonstrate with just pure water so you can see, okay? So we're back. I've got a test tube with some water and the tiny test tube. And what you got to do is fill the tiny test tube almost to the top, like so. And then what you're going to do is hinge them together like this, slowly but surely. And you're going to try to get the liquid in the big one to almost touch the liquid in the tiny one. And when you get close enough, you just throw it in there like that. And if you look, there's no bubbles whatsoever inside there. If you happen to screw it up, a lot of times you'll get this big old air pocket. But if you got quick hands like that, you can almost guarantee you won't get in there. So if I just do this, you can see there's a giant air bubble in that tiny test tube, and that's not what we want, because we want the CO2 to collect in there. So if you have a test or a bubble in there, that could be a potential error, okay? Okay, so now I've got them all the yeast in it and all the tiny test tubes, and I made sure that there was no bubbles in the tiny test tubes as I inverted them. There was a couple that I had small ones, so I had to tip them up, turn around, to make sure that all that was good. Okay, so now what we're gonna do is we're gonna put them into the incubator, which is gonna be in Mrs. Coffee's room. It looks like a giant refrigerator, but instead of producing cool air, it produces heat. So here you go. Okay, so those test tubes are going to sit in there for 24 hours at 30 degrees Celsius, and that temperature will hopefully activate the yeast to be an ideal temperature range for them, so they can munch away on those sugars, turn it into carbon dioxide, and hopefully we'll have some data tomorrow. Thanks for tuning in, guys.